One of the earliest attempts to model human population growth by means of the mathematics was by Thomas Malthus in 1798. The idea behind the Malthusian model is the assumption that rate at which population of a country grows at a certain time is proportional to the total of population of the country at time. The experiment consists of verifying that the bacterial growth is given by a differential equation, with the aim of verifying that the experimental results are similar to those given solving the differential equation with the initial conditions assumed by the experimental results.
Our schematic representation of the bacterial culture petri dish is that everything had to be sterilized. Then the sample was taken from the tonsils called throat swab culture. Then we need a nutrient medium that was an agarose gel. Then the petri dishes were incubated at the best temperature for the growing. And finally, we use an spectrometer to know the spectral absorbance. And this is the procedure. With absorbance of each culture, we can know the number of bacteria in this with turbidity method.
Here you can see the differential equation. We solve it by separation of variables. You can see the procedure below. Also the characterization with the initial conditions given by the experimental part, and also the Euler transformation and its characterization of two examples and both graphics. Conclusions. This simple model, which fails to take into account many factors that can influence human population to either grow or decline, nevertheless is still used to model growth of some small population over short intervals of time. We have found approximate values given by the differential equation. However, we conclude that the changes are caused by several circumstances affecting population growth of bacteria. For example, the type of bacteria, glucose, pressure conditions. Although we have controlled temperature and food, many other factors are involved.